Welcome! In this video tutorial, I will briefly show you how to organize your data to use Advanced Cell Classifier. I'm going to describe the most common structures supported by Advanced Cell Classifier, called standard screening structures based on image and feature files. However, other data structures are supported as well. Before running Advanced Cell Classifier, please read the help documentation file of the software. Advanced Cell Classifier requires an input dataset that is organized into a hierarchy of 3 levels of folders called Project Folder, Plate Folder, and Data Folder. The first level, Project Folder, is the main folder. It can contain any subfolders called plate folders, where each represents one plate of the dataset. The names of the plate folders are used internally by Advanced Cell Classifier as the names of the plates. Each plate folder must contain 3 subfolders called Contour Folder, Metadata Folder, and Original Image Folder. The Contour Folder contains segmented images. These are RGB images with contours highlighting individual objects. The image types supported by Advanced Cell Classifier are TIFF, BMP, PNG, JPG, and GIF. If sub-objects or subcellular compartments are visualized, we recommend coloring their contours using different color combinations. The metadata folder contains a meta file for each image, typically a TXT file with numerical values separated by a white space. A data file has as many rows as the number of cells present in the image and as many columns as the number of features computed for each cell, plus the spatial coordinates of each cell. The metadata folder must also contain a file named featurenames.acc reporting the headers of the numerical values stored in the text files. The headers are listed in different rows, and every header must be a single word without white spaces. The original image folder contains the original RGB images. Typically, the original RGB images present a gray level image in the blue channel referring to nuclear staining, for instance DAPI, an image in the green channel referring to GFP signal, and an image referring to RFB in the red channel, but this is absolutely arbitrary. Here you can see a sample dataset organized as described. The project folder is named Test Project Folder 01. It contains 2 plate folders named Test Plate Folder 01 and Test Plate Folder 02, and each of them contains ANAL1, the contour folder, ANAL2, the metadata folder, and ANAL3, the original image folder. Contour folder and original image folder contain images with the same names as the TXT files included in the metadata folder. Furthermore, the metadata folder also contains the featurenames.acc file. It is worth noting that image and metadata files must follow the naming convention of plate name, underscore, W, row and column of the well, underscore, S, a number identifying the image, etc. Note: plate name is the name of the plate. It must be the same name as used for the plate folder. The first character of the plate name must be a letter from A to Z. All the characters can be used for the plate name except underscore, asterisks, and white spaces. Please read the help documentation file provided with the software to find out all about the nomenclature rules. Finally, to open the dataset in Advanced Cell Classifier, simply click on File, New Project, then define the path to the project folder of the dataset, the names of Contour Folder, Metadata Folder, and Original Folder, and the other parameters of your data. Now you can start to analyze your images. Thank you for your attention.